Okay, so if you're just watching this recording, I had forgotten to press record. Sorry, guys. So we're in the middle of answering the how does B cell get activated, and we're redoing this little activity from the live stream session where we're putting these steps in order for how to activate a B cell. Okay, so I got at least one response in the chat, so we'll just look at it. And that's right, looks like y'all guys are all on the same page. So we start with antibody binding the antigen. That brings the antigen inside where it gets chopped into pieces, displayed in the MHC2. Although this step is not explicitly described in this activity, the CD4 on the surface of the T helper has to find the MHC2. That helps the T cell receptor bind the antigen inside of it. The T helper then activates the B cell with a cytokine emission. The B cell then proliferates, creates an army of itself, differentiates into plasma and memory, and the plasma produces lots of antibodies for now, and the memory saves it for later. Let's scooch on down to the next one that Sabrina asked about, which is how do T cytotoxics work? So they are scanning body cells. And for example, here's a body cell that's been infected with a virus. Endogenous antigens, everything that's made in-house, is displayed every day on these MHC1s. The T cytotoxic has a CD8. It helps it to find MHC1s. And then its TCR can check what's inside. And if it finds something inside, what it does is it releases perforin to poke a hole, granzyme to go in through that hole and initiate the apoptotic program of the target cell, leading to blebbing. And let's see. Does that answer your question, Sabrina, or do you have any follow-up questions? Okay, great. All right, if anyone else has a question, go ahead and use the reactions box to raise your hand or write your question into the chat. If both of those things are for some reason non-functional, we're a small enough group that you could also just unmute and state your question. Great. Glad to hear it, Sabrina. All right, what else is on your mind, guys? Was that the only thing? It's going to be a short day. If we have any requests, we could go over any of the Kahoots if you want to. We could really review anything you want, any calculations, concepts, whatever you need to feel ready for tomorrow's test. Roman, yes. So remember in this section, especially epidemiology, there's some simple fractions, right? And percentages, prevalence, incidence, mortality rate. In the live stream, we also learned how to calculate case fatality rate, although that won't be on the exam because it's not in the lectures. I just haven't had a chance to update it, but I will for next quarter. So if you're retaking the class, woohoo, you get to be tortured with yet another math. But yeah, the simple ones, right? So Stephanie asked, could we go over morbidity and mortality rate examples, please? Okay, yes, let me just pull up that one as well. And just to be very clear, what you'll be tested on, morbidity, you can calculate the morbidity rate, but because that is actually the same as prevalence, I'm not making it a separate thing. So morbidity just by itself, if you just see the word morbidity, it's a total case number. It's a raw number, like 1,721, right? It's going to be the total case number. That's morbidity. That's regardless of when people contracted a disease. And mortality will also be a raw number. Mortality is how many people have died. Mortality rate is how many people have died over the total population. It's kind of a metric of how likely it is that you are to die, like sort of as an average living in a particular population, let's say the US, or let's say the population of the students on campus or whatever. So it's different from the case fatality rate. Mortality rate is out of total population, regardless of whether you were ever infected. And case fatality rate is of the people who are actually infected, did they die? And that's the one you don't have to worry about for the exam, even though it was in the live stream curriculum. All right, so let me just pull up the correct slides and we'll just have a quick look at that and some of the examples. It'll just take me a moment. Okay. Like is the, there we go. So we'll just run through these concepts real quick. So morbidity again is total cases. So if I was to say in the year 2020, 1,112 people were HIV positive in a particular population, that would be the morbidity. It's not a fraction, it's not a percentage, it's just the total case number. These two metrics, incidence and prevalence, incidence is the fraction of population that has recently contracted a disease in some defined time period, like Tuesday or the month of May or something like that. So it's new cases over total population, and prevalence is total cases over total population. So that's how many people currently have it, and regardless of whether it's a new case or old case, or regardless of when they contracted it. And so we had this example, right? We had this example in the live stream session. So if you're coming from my online only sessions, this was an example where on a college campus of a total population of 30,000 students, there were 600 cases of the cold and 150 of those were new cases. So morbidity would be 600, incidence would be the new cases over the population times 100, right? Because you've got to make it a percentage. So this is the decimal fraction, and then if you multiply by 100, you get a percentage, 0.5%, and prevalence would be total cases over population, which gives you 2%. And then mortality is total deaths. Again, it's a raw number, but mortality rate is that raw number divided by the total population that you're considering. And then we also covered case fatality rate in the live stream session, but it will not be on the test because it's not in my canned lectures at this time. I haven't had a chance to update it. So the example that we used to calculate some of those things was, suppose that we were looking at the whole population of our county of Snohomish. And on this day, there was this many cases of COVID, and on the next day, there were this many, and this is how many people had died. How do we figure out all these things? Okay, so morbidity is going to be total cases.
And they basically,
which is
they don't
this.
want to like list it with someone else.
Mortality is going to be total deaths. Prevalence is going to be the total cases over population times 100 because you want a percentage.
I have been. Yeah.
Incidence for this particular example you had to calculate because you had to say, well, it's new cases. So I know what the old case number was. I know what the new case number is. So this is the difference divided by population times 100. And then mortality rate would be deaths over population times 100. And then CFR, again, not on the test, but just for funsies, would be deaths over caseload times 100. All right, so let me just ask you guys if that is clear, or if you want me to clarify anything related to that. So I'll just check the chat if this is like all good, or if you need any of these sort of revisited. All good, okay. And then Roman asked, will there be short answer questions on the test or only multiple choices like last time? There's a very small amount of short answer, and this is why. I need, for Unit 3, I need the turnaround on my grading to be extremely short so that you can have an answer no later than end of day Wednesday, so you can decide if you're taking the final exam. So most of the questions are auto-graded, but not all of them. So the format will be quite similar to previous tests, except less of them will involve any typing. There will still be the usual, right, where you have to fix a certain number of the falses from the true false section at the beginning. And other than that, I think there's just a very small number of short answers. Most of them are auto-graded, which is not to say that they're easy. You'll remember quite a few of the auto-gradeds from the previous tests had multiple drop downs to check off or matching and things like that. So it will still take you some time for those questions at the end of the test. And I believe my current version of the test, though I'm still messing with it, I think it's 48 questions. Okay. Sabrina says, what's the role of pattern recognition in the second line of defense? So there are a number of places where pattern recognition shows up, primarily in the complement activation. So you'll remember, and let me just see if I can pull up the correct, slides to kind of have those going while I'm talking. Let's see, we need innate immunity. So the complement is one place where it shows up, and that's part of the second line of defense. And that's primarily what we've talked about as pattern recognition. But there are a couple of other things I'll just mention, and then I'll go over the complement in more detail. In the adaptive immune section, we talked at the end about the natural killer cells. Natural killer cells are actually part of the innate immune response. And that's because they're non-specific. They're talked about in the adaptive immune response section because they derive from the same lineage as the lymphocytes, the Bs and the Ts, and they have the same tools for killing their targets as the T cytotoxics do, perforin and granzyme. But they're non-specific. Natural killers just look for cells that have the pattern of no MHC1. And those would be cells that would be missed by T cytotoxics. So they kill targets not because they know what's wrong with them, but because they know that they're misbehaving. That pattern is they're not putting out their MHC1. Similarly, the complement can be activated both by the adaptive immune response, antibodies bind antigens, that's very specific, or by patterns, primarily bacterial patterns. So the alternative complement activation pathway requires recognition by the BPD proteins of just bacterial carbohydrate lipid signature molecules, things that are typically found on bacteria. But the BPDs don't know which bacteria they're touching. They just know it's non-human bacterial. That's a pattern. The lectin pathway also binds to mannose, a sugar that's not found really on human cells. And so it's a pattern that's found on bacteria. And there's actually other ones we haven't gone into. And so you're not responsible for them. But there's patterns that initiate phagocytosis as well. And one that you sort of know is that when when anything is coated with antibodies or opsonized, that's a pattern that phagocytes recognize. They don't know what they're going after specifically. They don't know if it's a virus, a toxin, a bacterium, what strain of bacterium. They don't care. They just know there's a pattern. It's coated with antibodies. We need to get rid of it. So with that, I'll just quickly share the reminder of the complement, which is where we mostly focused on our pattern recognition, but it's by no means the only example of that. Here we go. Let's just go to the summary screen here. So again, the classical pathway, antibodies binding their antigens binding antibodies. That's a specific interaction that leads to this innate immune response. But both these pathways will turn on the complement because there's a protein made by the liver, lectin, that binds mannose, or BPD proteins that bind to carbohydrate lipids on the surface of bacteria. They're not specific, but that's still a pattern. So let's see if we can go to that slide. Here's the classical pathway. here's the alternative. So here's a bacterium. It's got a lipid carbohydrate complex on the surface that's non-human and sort of typical of bacteria. And these proteins recognize those types of signature molecules and immediately activate the complement. A pattern, not a specific. It doesn't know if this is E. coli, salmonella, staphylococcus. Doesn't, can't tell the difference. Similarly, the lectin pathway, this protein made by the liver called lectin binds to a carbohydrate containing mannose on the surface of bacteria. That's typically found on bacteria, not found on humans. A pattern, but not specific. Okay. So does that kind of help, Sabrina? Good. Roman says, what determines how long the memory cells last? Oh man, do the memory cells derived from T's or B's last longer? You know, it seems to vary a bit, and I'm not entirely sure. I would actually have to go talk to people who are kind of like at the front line of this field and say, hey, what do we know about this right now? And they might be like, well, that's a good question. It's unclear, and it seems to differ for different antigens. And I'm not sure. I can wildly speculate, and you don't have to take this to heart at all. It's not going to be on the test. But I think certain antigens have a stronger immune response, maybe because the fit between the antibody and the antigen is much stronger. Like what the B cells sort of are shuffled to, randomly generated to go after, they have no idea what they're really going to find. And so they might be looking for something, and they might be able to find, if they, whatever their recognition or their specificity is for, it might actually be able to recognize, like, say 2 or 3 things, but one of those things might be a much better fit. And so it's more often going to activate the B cell and maybe stronger, you know, so then you might get a stronger response from that particular antigen than another one that would still activate that same B cell because it was similar, but not quite exactly the same. I think whole agents tend to, because they have multiple epitopes, they're likely to trigger many, many B cells. And so you have a much, because the longevity of the B's and T memory cells is somewhat, I don't want to say random, but it's going to be a little bit variable. Sort of like by accident, they might, just like you have some genetic tendency to live so long, but you might get mowed down by a bus tomorrow, right? So the cells in your body kind of also encounter reactive oxygen species somewhat randomly and other toxins and things. So their lifespan isn't like predetermined for sure. It's just a tendency. So if you have lots of B cells and T cells activated because you gave someone a whole agent that's a really good mimic of the real pathogen, you're likely to have much longer lived populations just randomly, you know? Whereas if it's like a subunit vaccine, it's only one antigen and it's kind of shitty, but it's the best we can do, you're likely to have a shorter lived one. But truly, like, all I can say is that lots of people are working on this, and it seems to vary for different antigens. Think about how you have to have tetanus every 10 years, you know, like that's just sort of an average of how long do people's memory cells usually last for that particular toxoid vaccine. So it's an excellent question without a good answer, but luckily it won't be on the test. So does that help, Roman? As to your question, whether B's or T memory cells last longer, I actually don't know. And again, I'd have to talk to someone who is more an expert, like closer to the source of info and to the frontline cutting edge info that we're getting, because it's a very active area of investigation. Julia says, could you talk about the cells that can directly kill pathogens? So that would be primarily phagocytes and natural killer cells and T cytotoxics-ish. T cytotoxics, of course, are going to go after body cells, but some of those body cells one might consider pathogenic if they have been converted to a virally infected or a cancer cell. You know, at that point, you're kind of like, you're on the other side, you're the enemy now. So the way that a T cytotoxic cell works, as you recall, is that it finds some weird antigen and then it pokes a hole, perforin, and sends granzyme in to initiate apoptosis. Natural killers use those same tools. They just look for cells that are bare, and they're also effective. Natural killers are also effective against parasites, non-human parasites. And the majority of the other events would be phagocytic members of the innate immune response, the second line of defense. So that would be dendritic cells. They are not primarily the ones that kill everything, but they're scavenging things, and they might kill something along the way to find out what it is. Macrophages and neutrophils are going to be the primary agents of killing pathogenic cells like bacteria. And there's the parasites will be attacked on the exterior by natural killers and eosinophils primarily. So does that answer your question, Julia? Yeah, great, good question.
Yeah, can you hear me?
I can.
Yes, I was kind of confused because the dendritic cell, because I know that's phagocytes, but I also, it says like they present. So they also, APC,
Yes,
in presenting cells. So I was kind of confused, like.
Yeah, so they have this like really interesting role. They go around the body, they just find whatever they can, and they take it inside and present this. So their primary role is to provide information to the adaptive immune response by talking to T helpers about what they have found. Right? They're scouting the body, they're sort of running all around the body. And a lot of times the things they're finding are already dead, like pieces of bacteria or a toxin or a virus, which is not really technically alive. But if they were to find a bacterium, a whole bacterium, they could still potentially take it up and chop it into pieces. But their role, the reason that they would be doing that and sort of their effectiveness is not in primarily being responsible for clearing an infection. Their reason is there's something here, let me find out about it, kind of more scientific. Information gathering. And so they could still kill a cell doing that, but they're not going to be like when we talk about macrophages and neutrophils, we're talking about big armies of white blood cells coming to the scene because they know there's a problem and they know that they're supposed to kill something. They don't know what it is, but they've been told to go take care of business. And so there's lots of them. And so most of the pathogen removal is accomplished by those populations. The dendritic cell might kill one by accident just because like there's something, let me find out about it. Right? Does that make sense a little bit?
I was kind of, well, it's phagocytes, but they also present. So what is it? I mean, okay, alrighty.
Yeah. So just think about them as being like more like scouts. Like, if you have an army and the army is going to a battle, a lot of the army is composed of macrophages and neutrophils. But ahead of the army, you're going to send the scouts out, a few of them, just a few, to kind of find out what's going on. And one of the things they might find is some live enemy. Well, they might have to kill that live enemy to bring it back to camp to say what's going on. Does that make sense? But they're not like, there's not going to be enough of them to just take care of the whole problem. They're going to come back and say, this is what I found, talk to the T helpers. The T helpers are going to help sort of strategize. And meanwhile, the macrophages and neutrophils go in and clean up the problem.
Okay, thank you.
Okay. Neutrophils go after things that are opsonized and like coated with antibody or coated with that complement protein. And macrophages are activated by T helpers. So neutrophils are already active-ish, but they're very short-lived. And macrophages are inactive unless they are activated by a T helper, because the macrophage is quite damaging. If you let it wander around, it's just going to do all sorts of collateral damage. So you need it to be activated in order to sort of say, yes, we need you now. Now we don't stand down. We don't need you now. What are the ways that the innate and adaptive immune system communicate? Yes. Oh, so many ways. So your example, Jade, dendritic cells, which are innate, talking to T helpers, which are adaptive, right? So bringing intelligence back. The dendritic cell has no idea what it's bringing back. It's just blindly kind of gathering stuff up and taking it to the T helper and letting the T helper decide what to do about it. So that's an example. The complement activation. So when antibodies are made by the adaptive immune response in response to a specific threat, they go and coat whatever that threat is, a toxin, a bacterium, a viral particle. And then the phagocytes see that. They don't know what it is. They just see it's coated with antibodies. So they go chasing after it and gobble it up. So adaptive, innate, another conversation between the two of them. Let's see. Those are the best examples. And there are others, which I'm totally blanking on. If any of you can think of the ones that I actually talked about in class, you're welcome to put them into the chat to help us both out here. Let's see, I have a slide on that somewhere. Let me see if I can locate it. That's epidemiology. I don't want that. That is adaptive. Innate. Yeah, those are the two ones that are like, oh, collaboration. Here we go. Here we go. There's the slide I wanted. So complement activated by the classical pathway. That's a sort of classical example. Antibodies that coat targets to increase phagocytosis, and T helpers activating macrophages. So when a T helper knows something's out there, it will activate macrophages. So if a macrophage comes and says, hey, look at this, and the T helper looks at it and goes, oh man, that's a problem, it'll activate the macrophage to become more phagocytic. Okay. Jessica says, what type of vaccine would the Pfizer COVID vaccine fall under? This is such a fun question, Jessica. Isn't it fun to just like learn about stuff and then you're like, this is so topical?
I literally was like so into that part of the lecture. I was like texting people about it. I was texting my sister and I was like, oh my gosh, this is so cool. So I'm like totally interested.
Yeah, and you know what? Things have been moving so fast in the field that I actually haven't even had time to update my slides to answer that question, but I can answer it. And we talked about it in the live stream session for the live stream students. You'll remember this. So in the canned lectures, like up until last year, basically, the only nucleic acid vaccines really that were commonly available were the DNA West Nile virus one for horses. It was not a common vaccine for people at all. But the COVID-19 vaccines, Pfizer and Moderna, are both RNA based. So they're nucleic acid vaccines. And that's like basically the first like widespread use of nucleic acid vaccines that's effective in humans. I mean, people have done trials and stuff, but it's pretty amazing. Like it's revolutionary. Like so much stuff has happened this year that's hard to sort of like wrap your head around all of it and really appreciate all of it. But that is an amazing advance. Like if we continue to see good results from that and not like any weird long-term side effects, that will revolutionize vaccines, period. So yes, it's a nucleic acid vaccine. It's RNA based, which makes it different from the West Nile. And it's got a fatty lipid coating so that it gets into cells, and then the cells have the RNA, and then they make the antigen, and then the antigen causes an immune response.
Okay, yeah, because they were offering, so where I work at the hospital, they offered us the Pfizer one. And so I got the Pfizer one, and then I heard of other people getting the Moderna one, which for me, I didn't like, my immune response was like great. I mean, yeah, I felt like, that a little bit feverish, but like I was happy about that because that means like it triggered
It's working.
right. So like it's like, yeah, you kind of want that because it's like, it's doing something. And then other people like who got the Moderna were saying like, oh my gosh, they got really bad side effects like the fever, the chills, the night sweats and all that. But it's all telling you that it's working.
Yeah.
Like
it seems really variable. Like there's also quite a lot of evidence that typically older people don't have a strong overreaction, but we've got a faculty member. I was in a faculty meeting just a couple of days ago. She got just the first shot, and it's supposed to take that second dose before you get really serious side effects if you're going to get them. The first shot made her sick for 4 days, like off her feet, and she's a person who never goes down. So, I mean, it's just kind of all over the place. So Moderna and Pfizer are both RNA vaccines. They're very similar. The Johnson & Johnson one is actually an attenuated live, but it's not COVID, it's a adenovirus. So it's a different virus, and it's been engineered to express the glycoproteins of COVID. So while it is a live virus, it doesn't cause an active COVID infection, and it just has the antigens on the surface. So that's also, you know, a relatively new technology in the sense of having these like bioengineered viruses that are kind of a Frankenstein of an old virus and a new one. Sabrina said, no bad side effects, but the second dose triggered an asthma attack. Wow. That's crazy, huh? Yeah. So it seems to be pretty much all over the place, but I think it's fascinating. So yeah, those 3 ones out there, the 2 ones are RNA and the other one is a live virus, but it can't give you COVID because it's actually not a coronavirus. It's an adenovirus, a different virus altogether, just been changed so that it expresses the antigens, surface antigens look like COVID. Okay, next question, guys. What else is on your mind? I'm just checking the chat, make sure I didn't miss anything. Yeah, and Jessica, yes, just to reiterate, the nucleic acid vaccines never have live virus in them at all. They're sort of like they're not like a subunit, but they're like a subunit in a way, in the sense that you can't get the illness from it because it's like not the actual agent. Okay, so great question so far. What else is bubbling up in your mind that you need to know? If it's even off topic of Unit 3, you're welcome to ask those questions too. I'll just give you another minute to sort of ponder and let things bubble up. But if all your questions have been satisfied, in another minute we can end early if you want to. Awesome. Oh, can we go over exo versus endotoxin and LD50? Yes, absolutely. Give me just 1 second. That's going to be pathogenesis. So let's just say first off, yeah, you are not alone, Jessica. Other people are cheering on your question. They wanted that answer to that as well. So let's just go to endo versus exo. Here we go. This is a good slide, kind of summarizing the difference between the big difference between the categories. Endotoxin is a structural component of the extracellular, external leaflet of the gram-negative cell wall. It's called the lipopolysaccharide layer. So you'll remember there's like the membrane, the single layer of peptidoglycan, and then this external phospholipid bilayer. And the very outside leaflet, the half of that phospholipid bilayer that's on the outside, has a lipid in it, floating in it, called lipid A. That's an endotoxin, and it's attached to the A polysaccharide. So lipid A is an endotoxin. It primarily acts as a superantigen. So when you get exposed to it, it causes a overactivation of your innate immune response and inflammation. And if that's very severe, it can kill you. So it can range from like sort of mild systemic inflammation to you're dead. But because it's an endotoxin and lipid-based and not as potent as exotoxins, you need a pretty high dose of it to actually kill you. Most people just feel like sick. The other thing that's true about endotoxin is because it's a component of the cell wall, it is not exposed to you don't get exposed to it unless a cell is falling apart and little pieces are falling off. So when the cell starts to die, which is going to happen, right? Cells only live so long. So eventually one will start to die and then you'll be exposed to lipid A. But really, you're not going to be exposed to high levels of it unless you have a massive overgrowth of one particular species and they all kind of die at the same time, especially like if that'll happen if you start taking antibiotics and then they all get killed all at the same time. And so temporarily have kind of a endotoxin overload. Again, not usually enough to kill you, but you could feel a little shitty for a minute. All right, now exotoxins are proteins made by bacteria that go out, they're secreted, and they go off and do various things. There's 3 types, the AB type, the membrane type, and then the membrane disrupting type, and then the superantigen type. So let's have a look at the AB type. The AB type has 2 pieces. It's got the B part, which binds to the target, which is why these toxins can be very tissue specific. And then that causes internalization, as always happens with a ligand binding the receptor. Then it comes apart, and the second piece, the A piece, goes and usually inhibits the ribosome, so prevents protein synthesis, which may sound mild, but is really disastrous for the cell. The membrane disrupting types come in 2 forms. One of them just poke holes in cells. The other chops the phospholipid head groups off of phospholipids. Both of these cause quite a lot of damage. It basically destroys cell integrity. And then the superantigens cause your immune response to overactivate and a lot of inflammation usually. So there's sort of a lot of the conversations between white blood cells are accomplished by chemicals called cytokines, and the more cytokine chatter there is, the more chatter there is. It's like it's a positive feedback loop. So you can get to a point where you overexcite the immune response, and then the symptoms are very severe. It can range from resetting your body temperature, nausea, vomiting, diarrhea, to you go into shock and die. The other thing that was a good way to look at these is these comparisons. Exotoxins can be made by any cell type because they're just, instructions in the DNA to make whatever protein toxin. Because they're proteins, they're antigenic, so you can make an immune response against them. You can develop immunity, you can develop toxoid vaccines, you can post-exposure prophylaxis can be, you give someone passively antibodies against this, so they can be neutralized by antitoxin. And their LD50 is small because these are quite potent. You don't need very much in order to be affected. Whereas endotoxin, made of lipid, not antigenic, you don't develop immunity, you can't make a toxoid against it. It's not neutralized by antitoxins. It's not denatured by heat. So if you have food that is being cooked and there's already endotoxin in it, that's not going to remove the endotoxin, but it would remove the exotoxin, which is a protein that can be denatured. The LD50 is relatively large, which is good news. So even though you can't easily destroy it, you need quite a lot of it to hurt you. And I think that's pretty much it. Does that help to address, your question, Jessica? Great. Is there a way to get rid of it if heat doesn't affect it? The best way is to not have the food be infected with gram-negative bacteria in the first place. So what you'd want to do is have your natural ingredients, the raw foods, cook them, right? So usually if you rinse them well, they don't have a lot of bacteria in them except the surface. So you rinse them well, you cook them, the bacteria is gone. If there's a little bit of residual endotoxin in the raw food, it's not likely to hurt you. You cook it the first time, you've killed the bacteria, so they're not going to grow and form a large population. Remember that LD50 is large, so it would take a lot of endotoxin to hurt you. And then when that food is cooked, you then follow your 4-hour rule, right? You don't let it sit at room temperature longer than 4 hours. And so you don't get a large growth of bacteria falling in, or endospores germinating or whatever might happen. So by maintaining temperature control and then throwing it out after it's been in the fridge for a while, you can minimize the chances that a bacterium will grow and generate endotoxin. But what could happen? Let's suppose that you cook the food and then you let it sit out for more than 4 hours, and it got contaminated with some gram-negative bacteria, like E. coli or salmonella or something like that. And because it was sitting out, they had a chance to grow and grow and grow. Then you stick it in the fridge, and then you have it for breakfast on Tuesday or whatever. Then you could be exposing yourself. Even if you reheat it in the microwave, the endotoxin's already there now. Does that make sense? Yes, Jade. So usually, the antibiotics are going to kill the gram-negatives kind of at a, and more synchronously. But there's, again, the LD50 is working in your favor here. Probably you're not going to be exposed to such a high dose. Your body will be able to clear it and you'll feel better soon, but you will maybe feel bad for a minute when you take the drug. And then you just wait it out. Yeah. It's just something to be aware of. Like, if you had such a high load of gram-negative bacteria that the antibiotic would release enough endotoxin to kill you, probably what that means is you were done for anyway. That would be pretty high load of bacteria. They're doing all kinds of things to you at that point. So, I'm not a medical expert. So if you're in that situation, maybe don't listen to me. Talk to your doctor instead. But yeah, it's not going to be very common that you're really in danger, but you might not feel great. Okay, any other questions, guys? Any other questions, concerns, comments, feedback, burning need to knows? I'll just wait another 30 seconds to kind of let the reality of the situation wash over us. Any final questions come to bubbling up from your mind?
I actually have a question about the course evaluation, if I could ask that real quick.
Oh yeah, sure. And then Jessica asked a great question in the chat. So go ahead, Jade, ask your question.
So the course evaluation, the informal one that you want us to do, that's not the same as like the school's course evaluation, correct?
That's right. It is different.
Okay. Okay.
Did you get a formal course eval?
Don't we have to log in to something to do that? At the end of the quarter, we have to log in somewhere to do those.
Yeah, but they're not auto issued for every class. So
Oh, okay.
I usually get a heads up if my course is being formally evaluated. Since I'm more senior now, they used, like, if I was a rookie, they would evaluate me every quarter just to make sure I wasn't like completely damaging young minds. But but they've given me a certain level of a pass at this point. So you should, if they're going to formally evaluate me, usually I get a notification and then you guys would get one too. And
yeah, I haven't gotten one. It's just for almost like every other class that I've taken, there's been one. Mm-hmm. Even for teachers that have been there or professors that have been there for quite a while. But I was just curious. I wanted to make sure that it wasn't the same.
Yeah, it's different. It is different. So okay. If by chance you get a notification for the formal one or you log into that evaluation center and you find out there is one, just let me know. But okay. I don't think there is one this quarter, but the informal one is an opportunity for you to still give me feedback and get credit. And I really find that valuable because the formal ones I don't get to see until like 6 weeks after the quarter's over. So mm-hmm. that there's no, it's all anonymous and everything. But that means I don't have time to read them and go, oh, well, shoot, that was something I want to fix for next year. So I really appreciate this, like, more instant feedback. And so that's why I give extra credit to kind of like incentivize people to give me their honest opinion about things.
Okay, great. Thank you.
Thank you so much for asking that. Okay. So yes, just the informal eval is online. It's available. It's not part of the college at all. I do read it right away. I give credit for that. Question from Jessica. Low ID50 is more infectious. Yes. And then you said less likely to kill you, irrelevant. So ID50 and LD50 are just separate metrics altogether. There are some diseases that are extremely infectious and non-lethal. But in any case, LD50 measures the effectiveness of a toxin. Not all pathogens make toxins. And, you know, infectiveness has to do with how infective it is. And then LD50 has to do with how lethal it is. And they're really separate metrics altogether. So if you're asked a question about that, just know they're independent. They're not correlated in any way. Can you talk about the pathogenic properties of fungi and protozoa? Yeah, so most of the pathogens, like microbial pathogens, I should say, they're in that category of protozoa and fungi, do not make toxins. There are some exceptions. There's a few exceptions, but for the most part, they don't. A lot of the fungal toxins are made by those macroorganisms that are in the fungus kingdom. But the most of the microbial pathogenic fungi are not toxin producing. What they are is they make metabolic wastes. They often do a lot of tissue damage in the area that they're growing. And so that's the primary way that they harm you, or they can, you know, sometimes make proteins that are allergenic. They can cause you to have an allergic response. So yes. Okay, any other questions? Managed to kill almost an hour together. Good job, you guys. If anyone needs me to sing Baby Shark, I can do that. You know, I've got, I can also probably read from memory. The plot points to about a dozen baby books. It really did, Jade. The quarter went by in a blink of an eye. I don't even know what happened to the time. You know, it's because I was having so much fun with you guys. Yeah, I know. It was crazy fast. I don't know what happened. I feel like someone just turned the fast forward button on in the universal VCR machine.
I do have one question about the lab report.
Yeah, go ahead, Haley.
The due date for it. So in the notebook, should we put like that we started our lab report on this specific day and stuff?
Oh, no.
Get our
Yeah, the lab report should, or not, the lab notebook should be complete and accurate as far as the testing that you did, like, and all of the requests that you made to the lab technician and the interpretations and all that stuff. But once you're done, you wrote up, your last note would have been like, this test looks like this, it's red, it's negative, this eliminates the la la la, so this is my final species right here. And if you want to, if you did step 4, you could put your notes in there, although that's not required because step 4 is separate and it's extra credit and it's not required. So even if you did step 4, you don't have to put it in your lab notebook, but it does have to go in the drop box for extra credit, step 4. Okay. But at that point, your lab notebook is now done, unless in the course of writing up your lab report, you might be like, it's always possible. You're like, oh shoot, I made a mistake. So then you go back and you go, March 14th, I realized I screwed up the interpretation of blah, blah, blah test. So now I'm going to go reinterpret everything. You know, if that happens, that's what you add, but no, you don't have to talk about, the process of writing a lab report itself, the logistics of it. No. Does that help?
Yeah. And then also, is the extra credit one, was that still due last night, or did you extend that too?
The step 4, it was due last night. I mean, if you still want to do it today, the only problem is if you haven't already requested the DNA, like the tech's done dispensing things.
I did request the DNA. I just didn't have time to do it.
Yeah. I would still accept it until Tuesday for step 4 as
Okay.
well. Yeah, if you would do me a favor and shoot me an email, I can formally announce that.
Okay.
But again, just like the same caveat applies, like if you've submitted something to either of those drop boxes, unless you tell me otherwise, I am going to grade them and you do not get a regrade. So, because I've got to start grading today or I'm never going to make my deadline. What is the average score people get on that if they do it? Are you talking about step 4, Jessica? Oh, the lab report. So I guess I'm a little bit confused by the part that says if they do it, like everybody needs to do the lab report. I mean, if you don't do it, you get 0. But the lab report average score, gosh, oh, let's see. Typically people do fairly well, but it varies. Like, I've also had like, you know, I've had people get 10% on the lab report because they didn't do most of it. If you actually do all of it, you usually do relatively well, and you'll do really well if you follow the instructions and understood the process. So I don't know if that's very helpful. It's a pretty wide range, but the average is relatively high. I would say what would be a good, the labs themselves are graded more leniently. So you might not do as well. Like a lot of people are getting like 100% on the labs usually, as long as they're doing a decent job, like if you did it. So it's not like that. I actually grade it. I actually read everything. I see how well it was conceived and executed and interpreted and logically reasoned. But it's definitely going to be a higher grade than the exams for most people, unless you're a super good test taker. So it's sort of in between. I would say it's like usually kind of an average would be a little bit lower than what you're getting on labs, because labs are graded very leniently, and a little bit higher than what you've been doing on the exams, somewhere in there. How often do people get their species wrong? Huh. It's a good question too. You're asking like quantitative questions of someone who actually like is a little bit of a numbers person, but I haven't run the numbers, so I'm not sure. I would say, let me think. Since we switched to online, it's been higher because in the in-person class, you do the experiments yourself, so you can screw them up. And that doesn't really happen, except for every once in a while, the technician might give you the wrong result. But other than that, that's the only error that can be introduced. The other error, of course, is if you look at the test and you read it wrong or you misinterpret it. So 75% of the time people get it right, I would say, in the online forum. And I would say it's lower in an in-person class. It's much lower, actually. Your cat looks like, yes, and he's not just fluffy, he's very heavy. He's like a 16-pound cat, which is a big cat. All right. And let's see, I put when I started and finished in the lab report in the notebook. Oh, that's fine, Jade. Yeah, I just will ignore that, but it won't hurt you that you put in extra information. Oh, hi there, sweetie. Oh, you're so cutie. I'm talking to a cat. Those of you who may be listening at home, I'm like, please don't like get me fired for inappropriate behavior with students. That's a very cute looking cat. It's really a cat. Okay, now I'm like embarrassed. Yeah. Oh, is that cat like young, Sabrina? Is that a youngish cat? No, she's 6. Okay. This cat is like 13. He doesn't look it. He's he's aged well. Yeah, she is. She looks small. I have a small cat too. She's kind of shy. He's a massive, and I have this other cat who's small. All right, for those of you who are not cat aficionados and are not interested in sticking around for the Cat Fanciers Club, I could certainly talk about cats all day. And thank you for sharing your cat with me, Sabrina. That's very cute. But if you guys are done, you can go ahead and log out. If you have more questions, though, I'll stick around for a few minutes just to kind of mop up whatever is on your mind. And thanks for coming and asking such great questions. And thanks on behalf of the people watching at home who were not able to be here in person. I'm sure they appreciated all your thoughtful questions. All right. Thanks, guys. Have a good one. Good luck on the test.